And from out beyond the solar system, we're returning back into a focus closer to Earth and specifically on some of the life issues. SoRi La is an MSc candidate at the University of Witwatersrand, and she'll be speaking about the origin of eukaryotes and specifically the use of archaean genetic toolkits of programmed cell deaths to understand the origin of eukaryotes in a talk that has a lot to do with the early evolution of life here on Earth. Thank you.
Thank you. Good morning, everyone, or good evening. My name's SoRi, and I'm a master's student at the Evolutionary Studies Institute in South Africa. And first of all, I'd just like to thank you for letting me be a part of this amazing conference and present a field in the origin of life that was not focused as much until now, but it's vital to understanding how life on Earth originated and evolved, which is the role of death in the rise of eukaryotes. So because this is a multidisciplinary conference, I'm going to skip the technical details of my research as much as possible, but please feel more than welcome to ask me questions later. So in this presentation, I'm going to talk about a part of my master's project. And before I get into the talk, I would like to tell you the conclusion first so that you know what to expect. And I know it's not going to make much sense now, but it will at the end. So just bear with me, please. So the conclusion I got from my research project was that we found genes encoding the ancestors of one of the main effectors of programmed cell death in archaea. And this is actually quite an astonishing claim to make because, first of all, this suggests that ancestral programmed cell death toolkits were present since the beginning of early microbial life, whereas just a few years ago, programmed cell death was assumed to be a hallmark of multicellularity. But now we know that programmed cell death was very important for unicellular prokaryotes, where, for example, it can act as an altruistic mechanism to benefit the population. Secondly, we found that these ancestral death effectors were possibly associated with cell survival rather than programmed cell death at the point of the rise of eukaryotes. And this provides an interesting approach to the interconnected origin of eukaryotes and programmed cell death and how death was essential for the rise of complexity in microbial life. Okay, so getting back to the beginning, I'd like to set the scene as to where my research focus was in the history of life. And the period I'd like to focus on is this period where the first eukaryotic common ancestor, also called Fika, arose around 2.5 billion years ago, just after the rise of the two prokaryotic domains of life, the archaea and the bacteria, around 3.5. So here at the bottom, you can see the simplified hypothetical picture illustrating the concept of eukaryogenesis, the rise of eukaryotes. And it shows one of the most commonly accepted hypotheses of the rise of Fika through the endosymbiotic event between an archaeon and an alpha proteobacterium. And endosymbiosis, one of the proposed theories, involved a symbiotic relationship where one organism, suggested as the archaeon, engulfed the bacterium to produce over time a co-evolved relationship in which neither cell could survive alone. And obviously, there are many other models of eukaryogenesis, but they won't be discussed in this talk. So as shown on the figure here, it's expected that in a major transition of life, like eukaryogenesis, this symbiotic relationship would have constantly gone through cycles of cooperation, conflict, and conflict mediation for them to remain in a stable union. And one of the reasons of cooperation would have been metabolic complementation, where each other's metabolic waste would have been used. But conflict would have arisen due to their different evolutionary interests, such as the disagreement on efficiency of production waste. As closer the Earth got to the Great Oxidation Event, the oxygen level would have arisen, changing the environment that the organisms were in, and the aerobic alpha proteobacterium would have been in a more advantageous state and yielded higher energy than the archaeal host, which obviously led to problems. And for this conflict to be mediated, it seems PCD, programmed cell death, played an essential role as a mediator. And as a mediator, PCD would have promoted the death of the group. But in this talk, I'm not going to talk about why PCD was so important for eukaryogenesis, but rather I'm going to talk about a part of my master's project where I wanted to look at the evolutionary history of one of the main effectors of programmed cell death prior to and after eukaryogenesis, and how we can use this evolutionary history to study how PCD acted as a conflict mediator during eukaryogenesis. So to explain this a little further, this is a diagram of PCD. PCD is a genetically programmed form of cell death. And this generic figure shows the PCD molecular pathway in animals. And the reason I'm showing you this is to show how extremely complicated the molecular pathway of PCD is, involving multiple cascades of proteins and protein networks. But caspases are one of the main effectors in this pathway. And I'm going to focus on studying the evolutionary history of homologs of these caspases. So by homologs, I'm referring to proteins that have a common ancestor but can be different in structure and function. So to study the evolutionary history of caspases and its homologs, there were two issues that I initially wanted to address. And the first was the origin of eukaryotic caspases and caspase homologs from bacteria. And it's well established that eukaryotic caspases originate from bacteria. And this is because the mitochondria seen here, which originated from the alpha proteobacterium, played a major role in the activation of the PCD molecular pathway in coordination with caspases. And also, as seen on this figure here from an article by Aravind and Kunin, the phylogenetic reconstruction showed that the eukaryotic caspases clustered with the caspase homologs of various alpha proteobacteria, which further supports their bacterial origin. But the second issue that we actually don't know the answer of, and I wanted to address, was where did the genes in bacteria come from, and did archaea, the other prokaryotic domain of life, have any of them? So historically, archaea has been neglected from the PCD discussion because of the previous absence of any caspase homolog sequences in archaea. And this is especially due to the lack of genomic sequences available for archaeal organisms. As you can see on this figure here, there's a dramatic difference in the number of genome assemblies available according to GenBank. But advances in metagenomics, metaproteomics, and culturing methods are providing additional sequences every day. And the recent identification of novel archaeal lineages like the Ascot archaea, which is the closest superphylum to eukaryotes, was another incredible addition to the archaea research, as well as the first subcultured Ascot archaea, the Promethearchaeum syntrophicum. So with the additional data available, we wanted to know if we could find caspase homologs in archaea as well, and if some of the PCD genetic toolkits have a more ancestral origin than previously thought. So the hypotheses I was interested in testing was 1, caspase homologs are present in both bacteria and archaea, and 2, caspase homologs arose prior to the divergence of prokaryotes. For the methods, our approach was computational, and I'm not going to bore you with the details, and I'm going to move straight on to my results. First of all, we identified caspase homologs in archaea and confirmed our hypothesis 1 to be correct. Caspase homologs are present in both bacteria and archaea. So on the schematic tree of life with the universal common ancestor state at the bottom and the 3 domains. It depicts the taxonomic distribution of various caspase homologs and caspases in different phyla. The different caspase homologs include the metacaspases, the orthocaspases, and the paracaspases. So on this diagram, you can see that some of the caspase homologs, the orthocaspases and the metacaspases, are present in eukarya, archaea, and bacteria, including the deep branching phyla near the root. And it's important to know for later that only orthocaspases were found in this deep branching superphylum Depan. And Depan is just a superphylum of Archaea, and it's an abbreviation of the different phyla that's included in there. So this wide distribution of caspase homologs raised new questions and possibilities. Were caspase homologs the ancestors of one of the most important PCD effectors present in the ancestors of Archaea and bacteria, basically Lucas, or did they emerge after their divergence followed by horizontal gene transfer? So we wanted to take this question further and test our hypothesis 2, which stated that caspase homologs arose prior to the divergence of prokaryotes. Okay, so this is a gene tree of orthocaspases, and you can see the different colors representing the eukaryotic caspase homologs in yellow and orange, the archaea in pink, and the bacteria in blue did not cluster together. So the gene tree didn't correspond to the species tree, which should have looked like the tree of life shown before. And the most likely reason for the disagreement is horizontal gene transfer. Horizontal gene transfer, HGT, is the movement of genetic material between organisms other than by the vertical sexual transmission of DNA from parent to offspring. And it's an extremely common and an important mechanism in the evolution of prokaryotes. So previously I mentioned that only orthocaspases are present in deep branching archaea, and analyzing the taxonomic distribution with this gene tree, we came to a conclusion that 1, orthocaspases are the most ancestral form of all caspase homologs, and 2, the presence of orthocaspases in all of the major lineages of archaea and bacteria suggests they were present prior to the divergence of prokaryotes. Obviously, we don't actually have proof that these were present prior, and although we do agree that the tree is most fitting with HGT, we claim that presence of ancestral orthocaspases in organisms prior to prokaryotic diversification with HGT is the most parsimonious explanation. So we identified caspase homologs in archaea, and we're quite confident that they were present prior to the divergence of prokaryotes. But what does this mean? If these caspase homologs were present prior to eukaryogenesis, were they involved in programmed cell death or pro-survival? This is because I didn't mention this before, but this question arose because caspases and caspase homologs are pleiotropic. So this is yet again another complicated figure, but the main point is that the caspases over here act as the central mediators in PCD, but they also participate in multiple cell survival pathways, like cell proliferation and cell differentiation by activating different cascades of proteins. Therefore, we wanted to investigate the putative ancestral function of these archaeal caspase homologs, and how we wanted to do that was through phyletic pattern analysis, which refers to studying their protein domain and domain architecture to infer the ancestral function. So a quick introduction to part 2. This is a domain architecture diagram of caspases and caspase homologs. And a domain refers to distinct functional or structural units in a protein that is responsible for a particular function or interaction, contributing to the overall role of a protein. So in this diagram, you can see that caspases or caspase homologs all have the P20 domain, which is required for their proteolytic activity. But you can see here that for the caspases, they require additional domains like the DD to act as the receptors or the adapters of the cell signaling pathway. So on the diagram next door, you can see these additional domains that allow the extrinsic signal to be transferred from the death receptors to the caspases to allow PCD to occur in the cell. So it's extremely important for these domains to be there for these caspases to play a role in PCD. So I wanted to investigate what kind of protein domains archaeal caspase homologs possess to infer their ancestral functions. For the methods again, I am going to skip through this and go right into the results, where this is the domain architecture diagram of archaeal caspase homologs. And you can see here they all have the P20 domain as well as additional domains associated with different functions sorted by color. And their order is from left to right with their abundance on the side. So a lot of different protein domains were identified, including the death domains that are known to be associated with PCD. But you can see here that domains associated with cell survival, like cell proliferation and cell cycle, were more abundant. And even for the death domains that we found indicated in red here, a lot of other non-PCD roles were predicted, so it was difficult for us to confidently assume they were associated with PCD. So looking at the entire domain architecture of these proteins, we observed multiple caspase homologs indicated in blue here, resembling a signaling peptide responsible for interacting with the external environment and neighboring cells, as well as cytosolic proteins associated with cell survival. And this raised new questions about the putative role of archaeal caspase homologs during eukaryogenesis and whether they were once ancestral proteins associated with pro-survival functions. So to answer this question, the evolutionary history of different domains found in archaeal caspase homologs were mapped onto the tree of life. And obviously, we didn't look at the evolutionary history of domains of bacteria and eukaryote at all because we didn't do the analysis. So you can see here that for the orthocaspases here at the bottom, near the superphytum depan, possess no additional domains. But as metacaspases started to appear in phyla Thermoplasmatoda and Eukaryotes, they started to possess cell survival associated domains. And it's likely they were associated with some form of cell survival at this point in time. They also acquired additional sequence features for their regulation, like the calcium binding sites and the protein-rich repeats. And later, with the rise of the late branching TAC, caspase homologs possessing death domains started to appear. And at the point of eukaryogenesis, it's likely that archaeal caspase homologs may have played some important role. And just on the side note quickly, this evolutionary history actually supports one of the hypotheses on the origin of PCD, the original sin hypothesis by Mason. So some of the questions in this field is frequently answered in a philosophical nature, and a philosopher called Jean-Claude Mason suggested that PCD arose from a different combination and subsequent refinement of gene products participating in cell survival, and how we all have an intrinsic original sin in our genes to die. But going back to the original period of life we're focusing on, what does this mean for eukaryogenesis? So this evolutionary history has actually shown that there's an interconnected origin of death and complexity of microbial life. So this cycle of cooperation, conflict, and conflict mediation not only promoted the rise of eukaryotes, but allowed the PCD molecular pathway to evolve. So according to the original sin hypothesis, these symbionts were genetically primed to induce death in itself and in others. And with the rising conflicts, ancestral PCD genetic toolkits were incorporated into the conflict mediator mechanism for the rise of eukaryotes. But this pathway was also conserved and over time evolved into the PCD molecular pathway. So I'm going to try to explain this further with one of the most updated models of eukaryogenesis, which is the Entangle, Engulf, Endogenize E3 model by Imachi and colleagues. So this model states that eukaryotes evolved from a physical interaction and progressive internalization of an alpha proteobacterium inside an ascot archaeon through the initial cell projection mechanisms. So if I assume that Promethearchaeum syntrophicum as the archaeal host, I wanted to know the ancestral functions of the orthocaspase they possess during eukaryogenesis. And according to the domain architectures, this archaeon probably possessed cell surface and receptor-like orthocaspases associated with cell adhesion and projection, which further suggests they may be candidate PCD-related genes that were important acting on the surface of the cells. But this is a very early underdeveloped model of eukaryogenesis, and further analysis is required. So coming back to this conclusion slide I showed you at the beginning, I hope it makes more sense, and I explained how important it is when I say caspase homologs, the ancestors of one of the main effectors of PCD are present in archaea. And based on this data, caspase homologs may have emerged very early on, prior to the diversification of archaea and bacteria, and phyletic pattern analysis suggests they were survival related and later were incorporated into the molecular pathway of PCD. So basically what I'm trying to say is that some of the elements of PCD may be as old as the beginning of microbial life, where organisms were genetically primed to induce death in itself and in others. And I hope I've been a good enough advocate to show how death was essential for the rise of complexity in ancestral microbes, as shown by the suggested interconnected origin of eukaryotes and PCD. Lastly, I'd just like to thank these organizations for funding my research, as well as my supervisor, Prof. Durand, and my co-supervisor, Dr. Ndlovu, for their assistance. Thank you so much for listening to my talk.
Thank you very much. I'd like to open the floor then for questions, either from the other panelists or speakers or from the floor as well. Maybe I'll kick off with one, just sort of to try to make the connection over to some of the discussion that took place yesterday. Some of the things that have popped up around these issues relate to the possibility of places where life may have arisen. And we had some discussion yesterday even about evolving ideas about gradients, recognizing that there may be gradients necessary for the origin of life on the surface, but also in the subsurface. Is there anything about your findings that point us towards more or less likely settings or environments in which the eukaryotes might have evolved?
I'm sorry, but I think that's very far off from my scope of my research. I think it's hard to say. For my research, I know oxygen was important, but then again, to just infer the place of importance based on that, I think it's a difficult answer to say. Sorry.
No worries. Yeah, it was just to get the ball rolling. I didn't expect you to be able to actually tell us to get the discussion going. But do let me ask one quick related question. Do the levels of oxygen matter? I mean, one of the fascinating things about what was the great oxidation event is we now recognize that this is practically something that evolved with spatial and temporal difference. And we had places where we may have had microaerophilic environments or whiffs of oxygen long before we had the major oxygenation of the atmosphere. Do the levels of oxygen become an important aspect of some of the things you're discussing in terms of the rise of eukaryotes?
I'm also really not sure. I'm sorry.
No worries.
But I think it's not only oxygen that is important for eukaryogenesis. Obviously, there were many other factors. I was just stating one of the examples that was important at that time. But yeah, that's only what I can say, that it was oxygen itself was important.
Right. I see we have a question from Claudia here. Claudia, do you want to take the floor? You're still muted, Claudia. Is it just me? I can't.
No, she's muted.
Okay. If you'd like to try, if you can't succeed in having us hear you, do you want to try again? No, we're not getting sound. If you would like to maybe type the question, that would be fine, and then I can read it for you.
She is our next speaker too, so she'll.
Yeah, so you want to sort out. Wait. No.
So she can work on that, maybe.
Tell you what, Claudia, work on that. We have time, don't stress. But we do have a question. Thank you, Marshall. The question is, if life was evolving on Earth, Mars, maybe Venus, in the early solar system, was being shared by meteorites between those three bodies, could we detect that in the genetic data today?
Okay, so I hope I'm understanding the question properly. So you're asking if we can detect the conflict mediation in the genes in other planets, or is it only about ancestral microbes?
I think the question relates to the idea that meteorites might have in fact transported either material or life to Earth, Mars, and Venus together. And if that were the case, if there's a degree of either panspermia here or a joint origin, I hope I'm not putting words into Marshall's mouth. I think he's asking whether the genetic data in any way can help us resolve some of those long-standing questions.
I think genetic data definitely has the potential to. But regarding meteorites from other organisms, I really cannot say for sure how we would do that.
Right. And Marshall's just clarifying, the basic idea is if Earth, Mars, and Venus all had life and had a shared biology.
Yes, then I think there are many other examples of genes that did act as conflict mediator mechanisms. So if we could use examples of those genes to insinuate these mechanisms, then definitely. But I think this is really early research to say anything for sure.
Looks to me like we're getting Claudia back. Do you want to try your question again, Claudia, and use this as a sound check for your upcoming talk? Hey. No, we haven't got you. Sorry. Have to work on that in the background there. Any other questions from the floor or for the chat? I have one more I can slip in as well. One of the fascinating things, sorry, that just while we're waiting for other questions to come in, one of the fascinating things that those of us who work in sort of the deep subsurface microbial biosphere have found is that there are places where there are, in fact, eukaryotes, in particular things like nematodes. And I refer to the work of Gaetan Borgany in particular, who's identified nematodes in some of these deep subsurface sites, even as deep as a kilometer or more, where those nematodes are actually feeding on chemolithotrophic microbial communities. And again, I'm just curious whether there's any aspects of the results that you're getting that give us any insight into, have you taken a look at systems that are related specifically to subsurface eukaryotes, or is most of your work been focused primarily just on the surface eukaryotic biosphere?
So I've just been working on the subsurface and because it's hard to say because I've only worked on archaea and not on bacteria at all. So I am really not sure about every prokaryotic microbial system that's out there. So sorry.
No worries. I think people are just trying to go for some of the big questions here to think about. Now, you gave some spectacular results, and I think people are just curious to think about the broad implications of them. And so, for instance, I see a question from David Grinspoon, who is always good at those big questions. In replying to Marshall's original question, David says, it's interesting to think that these planets, and he's referring back then to Marshall's comment about the Earth, Mars, and Venus. David says, it's interesting to think that these planets may have had an intermittently shared biology, with perhaps occasional injections of organisms, and then long periods of isolated evolution. That might give them the challenge or the opportunity to co-evolve. And David wonders then what kind of genetic signature would then result from something like that.
I mean, there are already a lot of systems that like microbial systems that are existing in communities, not only in ancestral life, but in current modern microbial life as well. So I'm really not sure, but sorry, I'm really not sure.
Oh, no worries. No, like I said, these are just, I think people are very excited by your work and are therefore thinking about the broad implications. Yeah.
Yes.
Here's a more specific question coming from Pierre Schwab. Thank you, Pierre. Saying Scylla seems to distinguish apostosis from PCD. Can you give us a little bit more background for those of us who are not experts in this field about the difference between those two things?
So apoptosis is just a subtype of PCD, and apoptosis is very specific to complex eukaryotes like metazoa. So that's why when we study microbial PCD, we actually don't refer to apoptosis at all because we can't observe them in them at all. So the only link between microbial PCD to apoptosis that's found in like humans, for example, is like caspase and caspase homologs. That's an important link. Hopefully, I answered the question.
Okay. And then just looking to see whether we've got any other questions coming in here as we're got a few more minutes still that we can, particularly because we're having some issues with respect to hearing Claudia's. We do have a minute or two.
Maybe Claudia can try again.
More questions. Have we got sound?
Raising her hand.
All right, try again.
Can you hear me?
Yes.
So I don't know what's the problem with my microphone in my computer. So I'm sending my PDF slides for later to David because it's the only email I have.
Yeah, that's fine.
Great. And I can ask the question.
Please do.
Thanks. So I was just curious about your last slide, about your model, where you say that basically you have the bacteria that gets incorporated inside the archaea, right? So you will see in a moment, hopefully, that I work with lipids. And so I was quite interested in your model because we know that eukarya have lipid membranes that are made of components that are more similar to bacteria compared to archaea. I don't know if this question already happened while I was.
It has not.
So I was just curious.
So
I was just curious about how your model would justify the fact that eukarya have bacterial kind of lipids rather than archaeal.
Okay. So my model, so the E3 model is not my model. It's by the Imachi group. Yeah. So I just took that model into incorporating the PCD story because they refer to cell projection machineries. And it was just a suitable model for my analysis. But as for the cell membrane, I really can't say for that model, but I am aware of many other models that can be, but this was just the one that I chose. Yeah.
Thank you.
Thank you, Claudia. I'm glad you're with us now in terms of both sound and picture and slides, hopefully shortly. If somebody could maybe David make a final call on whether you want to go with Claudia next or Dietrich, and then pop that off to the hosted panelists. That would
Dietrich
be
Dietrich had to actually
we'll
teach
go with Dietrich.
for now. No, he has to teach, so we can go with Claudia.
So we'll stick
I have her
with Claudia.
slides.
Great.
Yeah, and I can present, and she can just tell me to advance.
Great. We've got one final question, I think, that's come up, and that's from Robin Catchpole. And again, just asking for a little bit more explanation about how PCD helps resolve conflict in the development of eukaryotes. Can you take a few moments to maybe just expand on that a little bit further? And just remembering we have such a breadth of different people online. It's all of us are learning so much.
Okay, so from my understanding, it's that, so if we have an early symbiotic group between an archaea and a bacterium trying to become an early feca, if there is a misalignment in their genomes or in their genetic aspects, then the entire the relationship would die. So both the archaea and the bacteria wouldn't end up surviving. So what I was trying to explain in the cycle was that, like, there were these different combinations of different bacteria and archaea coming together, and the ones that didn't complement each other perfectly would end up dying using a programmed pathway. And that's how conflict would be mediated for the future eukaryotes.